Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 7th October 2020. We were into the revision mode and we have been discussing about the principles of variation and inheritance, and in that we were talking about monohybrid cross, dihybrid cross, and I am sure with a lot of variations you must have practiced for the last 2 days, and I am expecting you to post your monohybrid as well as dihybrid cross practices with variations in our group. But so far I am yet to receive it. So please do post it because practice makes you perfect. Moving ahead, chromosomal theory of inheritance. We'll be talking about this. What is this chromosomal theory of inheritance? Inheritance, of course, is the acquiring characters through chromosomes or through genes from one generation to another generation. And now we are clubbing this, the inheritance, with that of Mendelian principles. Remember, three principles or laws we talked: law of dominance, law of segregation, law of independent assortment. So it is the union of this chromosomal segregation with Mendelian principles is called chromosomal theory of inheritance. So we need little bit of reflection on meiosis. Particularly in the meiosis 1, there are 5 substages, if you recollect from your memory, class 11th biology. Prophase 1, leptotene, zygote, pachytene, diplotin, dikinesis. Remember? And in that, the third subphase and fourth subphase, there will be a lot of crossing over. Chromatids will cross over. Also, you must remember here homologous chromosomes and heterologous chromosomes. Homologous chromosomes are chromosomes that look alike, that have similar structure. So in all respects they are same. Heterologous is different. The chromosomes are different. So homologous chromosomes having sister chromatids will have crossover, and that area in which the crossing over takes place is called synapse. So a lot of synaptic clefts will be formed during this prophase 1. So keeping that background in mind, let's move ahead. First point, chromosome as well as gene both occur in pairs. So there will be pairs of chromosomes and genes pair. We call it alleles. The two alleles of a gene pair are located on the same locus. Locus is position on a chromosome. And I said just now, homologous chromosomes. So where are the genes located? Two genes, they are called alleles. Alleles are present on the same position on a homologous chromosomes. So two scientists, we must be thankful for this, Sutton and Bowery, argued that pairing and separation of a pair of chromosomes would lead to segregation, separation of genes. So in the above statement, we are saying that two alleles are present together on a homologous. Now we are using the law of segregation of Mendelian, and then that through that we are saying that the two genes, that is alleles, will separate, and this separation will lead to variations. So Sutton united the knowledge of chromosomal separation with the Mendelian principles and called it as chromosomal theory of inheritance. So what does actually happen there? So you know, as I said, recollect prophase 1. So you got a G1 phase, growth 1, growth 2, and as I said, meiosis 1, particularly in anaphase, that is anaphase 1, we say bivalents will be formed. After crossing over, they get separated. So exchange of chromatids will take place. So through the exchange of chromatids, exchange of genes also will take place. And meiosis 2 further, anaphase 2, we are able to see the variants, and with the variants, with the recombinants, the germ cells will be having the chromosomes. These chromosomes which are present in the germ cells having varieties of alleles or chromosomes. So meiosis and germ cell formation in a cell with tetrads, four chromosomes. So linkage and recombination. When two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination was much higher than the non-parental type. Remember, yesterday we were talking about the dihybrid cross. In that we said 9:3:3:1. In that 9 is like as we said, we used one example, that is color seed, color of the seed, green or yellow, and nature of the seed, is it round seed or wrinkled seed. So one of the parental characteristic was round yellow, right? We said capital R, capital R, capital Y, capital Y. So here the proportion of parental gene combination was much higher than non-parental. Non-parental, that new combination, what is that? Instead of round yellow, wrinkled yellow is the new combination, right? Similarly, round yellow and round wrinkled, the two variations. So the new combinations would be, instead of round yellow, will have round green, and normal one is, another parent is wrinkled green. Instead of wrinkled green, round green. This is a new combination. So that is non-parental type. Morgan attributed this due to the physical association or the linkage. Why this round yellow numbers will be more? 9 we said. That is because of the linkage of the two genes, and coined the word linkage to describe physical association of genes on the same chromosome. That round and yellow numbers will be high. That is, parental gene combination is high because of the linkage. That is what the Morgan said. The generation of non-parental gene combination. Just now I said, what is that non-parental gene combination? What is parental gene combination? We said parental gene combination is capital R, capital R, capital Y, round yellow, right? Round yellow is the parental one of the parents, versus we said small r, small y, that is wrinkled green we said. This is the parental combination, wrinkled green. What is the new one? We said the new one is, this round will be there and this green will be there. So round green is the new one, and similarly wrinkled yellow, that's another new combination, wrinkled yellow. So these are the two, and this numbers will be maximum round yellow because of the linkage. The generation of non-parental gene combination during hybrid cross is called recombination. How is that this is possible? That is round green. Round green, how is it possible? Similarly, yellow wrinkled yellow, how is it possible? It is because of the recombination of the alleles or genes. So when genes are located on the same chromosome, they are tightly linked. So they are very much proximity. The locus makes the genes to be tightly linked and show the very recombination. So this is possible because of the crossing over. There is no doubt about it. So what is the difference? Crossing over leads to separation of linked genes, because once the sister chromatids cross each other, synaptic cleft will be there. After synapses, there is pulling of the chromatids, whereas the linkage keeps the genes together. And in crossing over, it involves exchange of segments between non-sister chromatids of homologous chromosomes, whereas linkage, it involves individual chromosomes. And in crossing over, the frequency of crossing over can never exceed 50%. The number of linkage group can never be more than haploid chromosome number. And it increases variability. Varieties are formed because of that, forming new gene combinations, whereas this linkage reduces variability because same combination will be there, right? So this is the difference between linkage and the crossing over. Now we go to another concept, moving at sex determination. How the sex of the individual will be decided. Henking in 1891 observed a trace of specific nuclear structure in few insects. He also observed that this specific nuclear structure is located on 50% of the sperms only. He called this as X body. He was not able to explain its significance. Obviously, it is 1891, remember? So that's how he could not explain. Later, it was observed that the ovum that received the sperms with X body become female and those not become males. So this X body was called as sex chromosome, and the other chromosomes are called autosomes and allosomes. In humans and other organisms, XY types of sex determination is seen. But in some insects like Drosophila, X0 type of sex determination is present. In both types of sex determination, male produce two different types of gametes, either with or without X chromosome, or some with X chromosome and some with Y chromosome. Such types of sex determination are called heterogametry. In birds, of course, ZW type is there instead of XY. ZW type of sex determination is present. Two different types of gametes are produced by females. Here see, there two different types of gametes are produced in male. Male produces two different because the sperm carrying X chromosome and the sperm carrying Y chromosome. So two different sperms will be there. But ovum has no choice. Ovum has got only one X chromosome. It doesn't have variety. Whereas in the case of birds, it is the two, as I said, the female will have that. So in case of men and other, female has no option. Female has no option. It has got only X chromosome. Whereas the male has got X as well as Y. Varieties will be there. In the case of birds, the female will have variety. That is, female will have both Z as well as W. The ovum will have Z chromosome and a W chromosome. This type of sex determination is female heterogametry. What did you say here? Male heterogametry. Here in the case of birds, it is female heterogametry. So sex determination in human beings is XY type. Out of 23 pairs of chromosomes, 23 pairs means 46 chromosomes. All of us will be having in every cell 46 chromosomes. 22 pairs, that is 44. 44 are autosomes. That is, they decide the characteristics of body. So autosomes are chromosomes that decide bodily characters. And remaining two, a pair of X chromosome is present in female. So you have 44 autosomes plus XX. This is the combination in the case of a female. And what about in male? In male, 44 autosomes will be there plus variety, that is XY. So presence of Y. If you see the difference between a male and female, in female it is 44 plus XX. In male it is 44 plus XY. So the difference is presence and absence of Y. If Y chromosome is present, it's a male. If Y chromosome is absent, it's a female. So during spermatogenesis, again I want you to recollect chapter number 3, spermatogenesis. Male produce two types of gametes or sperms. 50% carries Y chromosome and the remaining 50% contain X chromosome. Whereas female produce only one kind of gamete, that is ovum, having only X chromosome. So that's how the sex will be decided by the sperm. Is it the sperm having X chromosome or Y chromosome? So when sperm having Y chromosome, the sex of the baby is male. When the sperm carrying X chromosome fertilizes the egg, the baby is female. So it is the sperm that decides the sex of the baby. So here what we do, the parents having father first, the father being a male will be having 44 autosomes plus XY. mother also 44 autosomes plus XX. Now when division takes place, the gametes, and since it is meiosis, reduction will be there. So 44 will become 22, and out of the XY, one will have X, another will have Y. So 22 plus X one sperm, 22 plus Y another. In the case of mother, it is again 50%. So one will be having 22 plus X, another also it has no variety, no choice because XX 22 plus X only. So when combination takes place, for example, this 22 plus X and this 22 plus X, you get 44 plus XX. So having XX, it becomes a female. And this 22 plus X combines with another 22 plus X, 44 plus XX, again a female. Whereas a sperm carrying Y, when this combines 22 plus Y plus 22 plus X, 44 plus XY, this is male. And similarly 22, 22, 44 plus XY, male. That means you got 50% probability of a female child and 50% probability of the male child. So who is actually deciding the sex of the baby? It is the sperm carrying Y chromosome. So this is the deciding factor. If Y chromosome is present, it's a male. If Y chromosome is absent, it is a female. So there will be something called sudden changes will be there. That's called mutation. Mutation is a phenomena that results in alternation of DNA sequence and consequently results in change of genotype. And once there is a change in genotype, automatically phenotype also there will be change. The mutations that arise due to the change in single base pair of DNA are called point mutation. What is that single base pair? Like we said, remember, adenine and guanine and thymine and so AT and GC. If there is any change at one point of the nitrogen base, then we say point mutation. Like example, sickle cell anemia. So just keep this practicing and we'll move on to the next one, pedigree analysis. This pedigree analysis, there will be a chart also in related to practicals. So tomorrow in my class, we'll be discussing both the theory related to pedigree analysis and practical also, experiment also.